From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 2nd, 2023. Hello, Micronauts, and welcome to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 10th episode of Matters Microbial. Can you believe it? Thanks for listening and or watching. First, a member of my Wunderkammer. It's a little charm depicting the bacterium Delovibrio attacking and consuming another bacterium. I've long been interested in predatory microbes, and this makes me smile. I'll link to a wonderful review of predatory bacteria in the show notes. Today's inspirational lapel pins. We're in October, so this makes sense. The first is something from one of my favorite movies, 2001: A Space Odyssey. When Dave Bowman sees the giant monolith in orbit around Jupiter, he exclaims, "My God, it's full of stars," before being whisked away. The second inspirational lapel pin is from another favorite film of mine, the animated film The Iron Giant. The motto, "You are who you choose to be," is always with me. Incidentally, I was so delighted this morning to see Caitlin Carrico winning the Nobel Prize in Physiology and Medicine for her work on messenger RNA-based vaccines. My wife, Dr. Jennifer Quinn, created a lovely Lux portrait with luminous bacteria. It's so wonderful to see this happen. The 6th week of my microbiology class has been #microbial fun continuing. My Micronauts had a very successful set of PCR reactions from their water bottle buddy microbes isolated earlier, as you can see on this DNA gel. Next step, send them off for sequencing and compare 16S ribosomal RNA sequences to find out who's there. Next, my Micronauts have followed up on their marine microbiological safari. Most students were able to isolate typical marine bacteria from their marine samples, seawater samples, as you can see here. We were, of course, more interested in agar-pitting microbes, as you might see here. We even found a pitter with very dark pigment. But one Micronaut found a very wonderful fluorescent microbe, as you can see here. Next step, colony PCR and analysis to determine who each isolate is. Plus, one Micronaut was amusing with the pitter search by invoking Brad Pitt. Lastly, we're searching for antibiotic-producing microbes in lichen, following the guide of Joe Handelsman's quite wonderful student crowdsourced program, Tiny Earth. I will put a link to that impressive program in the show notes. Why lichens? Well, I hypothesize the lichens restrict what microbes can grow nearby as the lichen develops, so it might be a good source of antibiotic producers. My Micronauts collected lichens such as you see here, plated out dilutions in R2A medium. Very interesting microbial denizens were found, including some fluorescent ones, as you see. Our goal? Test candidates that might have antibiotic activity, as you can see here. It's always a little hard to identify them, but we work on it. And then we go further with 16S analysis. There are many important shifting paradigms in microbiology as it develops as a field, and it's always a challenge to bring students up to date on them. The great Elio Schechter has a wonderful essay on this topic, linked in the show notes. One of those increasingly important paradigms is called sociomicrobiology, how microbes interact with one another. While some scientists view interactions solely as positive, there are definitely microbes who compete with one another or even have more nefarious goals in mind. And who better to tell us about Microbial Fight Club than Dr. David Baltrus of the School of Plant Sciences at the University of Arizona, and a good friend. Welcome to our Quality Quorum, Dave.
Thanks for having me. It's great to be on the 10th episode, so hopefully many more following this.
Well, we're working hard on it, and I certainly have fun. Plus, it's therapeutic to hear from people doing wonderful microbial work like yourself. Would you mind telling everyone a little bit about, you're at what's called an R01 institution that produces PhDs. Can you tell the listeners and or watchers a little bit about how you conduct research in the lab?
Yeah, so we have a variety of different projects we work on, and largely I try to work with everybody that I can give an opportunity to. And so part of what drew me to this institution, but also to how I run my lab, is I didn't necessarily have the most greatest opportunity to do undergraduate research when I was an undergraduate, and so I tried to both bring in a bunch of undergraduates into the lab. There's, I think, 5 in there right now, plating out various different things. But also we have PhD students. So there are currently 5 or so PhD students, and then a couple of technicians too that that help me out with things. And then every once in a while, we'll bring in people that are called postdoctoral fellows that have gotten their PhDs usually somewhere else, and then come in and I try to help them create their own projects and kind of discover their own directions for research. So all sorts of different career stages of researchers that are going in the lab.
Well, I've known you for far too many years, sir. And I've got to tell you, you strike me as a really fine mentor, because you certainly have been a fine mentor to me.
Oh, thank you much. I try to do my best. That's right. We just try to do our best. So
that's all anyone can do, I think. So today's topic, Microbial Fight Club, and I hope everyone enjoys the artwork that my wife, Dr. Jennifer Quinn, created for this segment, which I thought was wonderful. Do you need a poster of that, Dave?
I would like a poster. That would be good.
So why don't you tell folks a little bit about your work suggesting that microbes can kind of fight one another?
Yeah, and so really this comes about from a variety of different types of research that we were doing. My lab is, I'm in the fun position of being able to ask questions for whatever curiosity that whatever direction we go in, and whatever we can make work with current funding rates. So the projects that I'm really working on now started probably about 10 years ago. We had a postdoc come into the lab named Dr. Kevin Hockett, who's now at Penn State University. Kevin was interested in he started off being interested in phage and how phage can interact with different bacteria and be transferred across different bacteria. And so we largely work with a lot of environmental pseudomonads, and so those fluorescent bacteria that you're probably finding in your water bottles. I have a large collection of them in my freezer, and so Kevin really just started off pitting them against each other. So we're like, all right, if we can find phage activity and induce it, let's see who can kill who, or where we can actually induce phage activity and see the results from that. This is a story in how research develops, though, because we didn't actually find a lot of phage activity. What we actually found was a lot of the strains were able to kill each other with bacteriocins. And so we stumbled into it not expecting to go in that direction, but we found this new killing activity that we then kind of dove into the literature and tried to figure out what was going on behind it. And so it ends up being a really cool evolutionary story and a really cool genetic story too, because what we discovered in Pseudomonas syringae, which is normally a plant pathogen found pretty much everywhere in the environment, but normally known as a plant pathogen, what we found is that they have co-opted through evolution prophage, so phage that have incorporated into the genomes of the bacteria. The bacteria over time, the genes within the genome that encode for phage capsids and phage replication have been lost. And so what's left is just a phage tail, just the end of the phage. And so the bacteria have co-opted this to, we think, kill competitors for, whatever they might be doing out in nature for competition. And so typically a single cell will produce maybe 100 or 200 of these phage tail compounds. Will lyse, and so there's a lysis module linked in a regulatory way with the production of the molecules, and so the bacteria lyse, and then they just kind of release them into the environment. And there's a couple of papers, not from our lab, but from other labs that have really cool movies showing it's really just explosive lysis where the phage molecules go everywhere. They make it out there into the environment, and then because they're proteins and it's really just a phage tail that's hanging out in the environment, they're pretty durable. And so we think they last for a decent amount of time. And we're not quite sure the evolutionary reasons why they do this, or whether it's for defense or for antagonism or what's going on there. But it's really cool. And I had no clue that these were created until we kind of stumbled into it. There's a much longer history of what are called phage-derived bacteriocins or tailocins. They were discovered in Pseudomonas aeruginosa, so a common opportunistic human pathogen. They were discovered probably 5 or 6 decades ago, and then characterized pretty well. We found them in Pseudomonas syringae and didn't expect them because we looked in the genome and we didn't see any of the known Pseudomonas aeruginosa tailocin genes there. So what we ended up finding and why we were able to characterize a kind of a new, completely novel source of these was that Pseudomonas syringae seems to have independently co-opted a different phage than the aeruginosa pyacins. They used to be known as the R and F type pyacins and aeruginosa. Pseudomonas syringae has co-opted a different phage, and so the genes are completely different. It's the same functions of genes, but from a completely different source phage. And so, for the strain that we found them in, we had the genome for maybe 15, 20 years, and it was always just a region of the genome labels it as a phage. It was there's a phage there. But when we actually found the activity, went back in and characterized it, we saw that it was a kind of an independently evolved tailocin within these strains. And so we had the idea, so we saw the killing activity, and then we had the idea that, well, if they are protein-based, they're going to be pretty durable molecules. They're non-replicative, so there's a lot of interest in things like phage therapy now, but these tailocins don't get out in the world and replicate. They're just there and mediate their killing activity. And so we've kind of gone down the direction of trying to figure out if we can use these as agricultural antibiotics. And so can we take these molecules, can we spray them on plants, and do they prevent plant infection? And so we have a couple of papers now showing that in the lab, under very controlled conditions, we can actually use these as agricultural prophylactic antimicrobials that can basically prevent disease from plant pathogens. And then we have a few that target things like E. coli 0157H7 or some of the Salmonella strains that we're working with. So we're hoping that maybe we have every once in a while in Yuma, Arizona, where it's pretty much any leafy green vegetable that you get in the winter in the United States is probably grown in Yuma, Arizona. Every once in a while, there's a outbreak of some nasty, usually E. coli or Salmonella that comes from those. We're hoping that we can develop something that we can spray on, something in the field to maybe prevent those E. coli and Salmonella from infecting the plants in the field. So that's kind of where we're going with it. I never think of myself as an applied researcher, but we just get to be curious and ask questions and figure out what's going on behind things.
Well, that's a wonderful thumbnail sketch. Do you mind if I ask a few questions?
Go for it, yeah.
So the first thing that occurred to me as you talked about this, are you aware of the story in Della Vibrio when the genome sequence first came out? Where I had a criticism of the paper, which I reviewed. It's been long enough I can say this. And they made a comment in the paper that Della Vibrio that lives within the periplasm, between the outer and the inner membrane of gram-negatives, some folks had thought that perhaps they were able to translocate ATP from their host. But the authors of the paper said that there was no gene related to the ATP translocases from Rickettsia or chlamydia, both of which do that. And I thought it was a strange thing for them to say because there had been work done by Ned Ruby 10 or 15 years before showing multiple ATP translocases in Della Vibrio. So what this reminded me, and it's something I try to teach my students, is yes, comparing gene sequences is great, but you only find relatives of what you already knew. And you're giving another really good example of that, I think.
Yeah, it was completely unanticipated and one where we try to set up experiments in the lab where you have things controlled in a way that you can interpret. And over and over again, I don't know whether this is just how I kind of run my career and how my career has worked, we keep stumbling into, that colony looks weird, or that's a weird result. And hopefully, we've controlled things in a way that we know, all right, it's a weird result, but it's a real result. So, can we dive in and figure out what the basis of this is? And we've done this over and over again, and that's kind of how some researchers are very focused on what they do. I'm just interested in finding the interesting offshoots of things that we're doing already and kind of developing them into new directions. So it's worked so far.
This is the Isaac Asimov quote that science doesn't advance by, he used to say, a damp Greek emerging from the bathtub shouting Eureka. Archimedes, I guess. Where it really advances is where somebody looks at what they're doing in the lab and says, that's weird. And that's 100% true. And you've given me yet more ammunition for that. By the way, one of the things I tried to do many years ago is I have a collaboration where we look at some of the microbes that live within the cloaca of lizards. And I would try to kind of battle against them. And I found many that were inducing phages in the sense that I saw, little plaques, but it doesn't have to be plaques anymore. And I think this is important for the listeners and viewers to understand, is that we're used to the idea of phage therapy, but the hand of Darwin's on everything. And using elements of the bacteriophage the way you just described as a tool, it's a wonderful example of natural selection.
Yeah, we've actually recently been lucky enough to, again, stumble into new results using more comparative genomics techniques. Basically, we have enough genome sequences now, and we have enough of these interaction matrices where we can just throw them into the computer and have the computer tell us about the correlations. And one nice, beautiful result popped out in that it looks like, and again, I should have read the older literature and kind of and seen this before with the piacins, but our Pseudomonas syringae talasins seem to be targeting the LPS, and so it's led down a new direction, lipopolysaccharide layer, which is sugars hanging off of the outside of the bacteria. So it's led us down a new direction of LPS production in Pseudomonas syringae, and we can use the talasins as a tool to understand the genetic basis of it. And it looks like, at least for Pseudomonas syringae, this is another one of those stories where if you look in the genome, all right, there's genes there that could look for, they could modify the LPS, they could do things in that way. But all of these enzymes are things like sugar transferases, and they're kind of generically annotated as, this one puts a sugar and takes it and puts it somewhere else. We're actually using the talasins to figure out a lot more of the genetic basis of these now, and syringae does the LPS in a different way, I think, than a lot of other gram-negative bacteria, in that it only has one variant of the LPS. Usually there's multiple chains that hang off. Syringae seems like it only has one type, and then different strains of syringae will completely swap out that one type for another type. And it's not to get too much into the details, but one chain is based off of D-rhamnose, which is what a lot of the other LPS is based off of. But then syringae also has a main chain based off of L-rhamnose too. Completely different interaction with the talasins, but just with that chimeric switch of the sugar, which is really cool. It's complicated to LPS chemistry is very complicated and is out of my depth. But it's been fun to kind of dive in and 30 years ago, LPS was chemically characterized in a variety of strains, and now we're on a hunt to try to find all of those strains and bring them in already, because we have the genomic resources to try to look at the underlying genetic basis of those things now, which is kind of cool.
Oh, I can't agree more. Please refresh my memory. It sounded to me like you were saying that when one of the bacteria bursts and release the talasins, this is how that's taking place. So it's almost like a group behavior or an altruistic behavior by microbes.
Yeah, it's I'm not going to attribute intent to it, because I'm an evolutionary biologist and we try to not do that too much. So a single strain will make about 100, 200, 300 of these molecules, and it has to lyse to release them. And so the single strain has to blow, the single cell has to blow up basically to release them. The way my hand wavy, what are these doing out in nature example of this, tying in a little bit more of our understanding of the regulatory nature of it, is the pathway that gets triggered to turn these on is through DNA damage. And so it's the typical SOS response. There's a repressor that is responsive to DNA damage that de-represses when the DNA damage is sensed, and then the whole pathway gets turned on. Once the pathway gets turned on, it's kind of set down to making the telasins, and then the last couple genes are they are lysozymes basically. And so it's, once that gets turned on, the cell is done for, and it's just a matter of how long it takes to get to that point before it blows up. What I think might be happening in nature is a lot of these environmental bacteria that make telasins typically are found in biofilms. So they're kind of in their little biofilms with their very closely related strains. But as part of, if you actually look into a biofilm, there's a lot of diversity in terms of what the cells are doing within a biofilm. And if you look closely, there's always a bunch of cells that are turning on the DNA damage responses for whatever reason. So I just kind of hand-wavingly like to imagine that that as they're parts of biofilm, maybe the DNA damage response gets turned on. They make these telasins, which are pretty durable protein molecules, and then it just kind of seeds the biofilm to prevent competitors from coming in. I'm going to work on that in the future to figure out if that's actually what's going on, but that's kind of what makes sense in my head for what these are doing in nature.
I like that idea a lot, actually, because, and again, I know it's not a matter of intent. I realize that, but, a microbial perspective is important, and we tend to be very reductionist, and that's not really how nature works. So it's curious to me. I mean, I think about, von Cooper's work with the mat colonizers, and you have competition within the mat. And it's always fun to talk about that with students. And this is another example. So what kind of microbial sense does it make in terms of making it to the next generation and prospering? So this is very interesting. I'm glad you clarified that. It's also true that there are all these different mechanisms where one bacterium can attempt to harm another. Not all bacteriocins, from my understanding anyway, are related to phage tails, right?
Yeah, there's a variety of other types that are made. I would say that the phage tails are a subset of the larger bacteriocins in general. There's a lot more that are known. A lot of them need to be internalized into the cell to actually have their antimicrobial activity, and so they'll get transported into the cell through various types of receptors on the outer membrane. The protein gets brought into the cell, and then typically they're acting through DNase or tRNase or RNase activity there. The phage tails are different than that because they don't need to be internalized. We think they just need to attach to the cells, and then they just put their spike protein through and lyse the cells, which kind of makes them a different class of antimicrobials than a lot of other things as well, which is potentially of use in going forward. So.
Oh, no, this is very interesting to me because when I show students, for example, the type 6 secretory system, and I flippantly call it a switchblade knife, but it sort of is. This is a more subtle version of that kind of thing. I especially like the concept that they have to be released by cells that have perished, and this then gets into the environment and acts as a way of fighting. Is it possible to take a bacterium which is currently sensitive to a talasin and make it resistant to talisins?
Yeah, so we've worked on this a lot too. It's driven us in a couple of interesting directions in terms of the LPS. And so when we actually look at one of the things we do is we screen for resistance in nature, and we look for strains that are inherently resistant. We try to screen for mutants that are more resistant to the talisins, and then we look for things like persister cells, which are not genetically resistant but can tolerate antimicrobials. And so the interesting part about talisins compared to other types of bacteriocins and other antimicrobials in general is that pretty much all the resistance we see comes with a huge cost in that the resistant cells have to shift their LPS, and the LPS ends up being a really critical interface between the environment and the bacteria. If that gets changed at all, typically there's a lot of knock-on effects for physiology of the cell, and a lot of different things can happen. And so pretty much all of the mutants that we see showing up when we just do the plating in the lab they are really sick. They don't grow really well. They're what are known as rough colonies, which are they'll grow on a plate okay, but if you put them in the environment, they don't do so well. So that's what we see genetically when we actually screen for them. In nature, bacteria are amazing and evolution is amazing. And so there's a variety of ways to become resistant, not necessarily through losing the LPS, but through bringing in new enzymes or bringing in enzymes that can manipulate the LPS in different ways to shield it from the talisin production or shield it from the talisin killing. And so my guess is that's going on as well. In Pseudomonas syringae, again, we've also seen that they seem to, and we don't know how this happens yet, but they seem to be able to completely swap out types of LPS through recombination, through horizontal gene transfer with other strains to completely reshape their LPS to become resistant to a previous version of the talisin. And then the last part is I mentioned persister cells. So we've seen a little bit of persisters in terms of talisins. There might need to be active growth for the cells to actually be killed by talisins. And so we've got one manuscript that looks at persisters and showing that basically they do exist, and maybe about 5-10% of the population might be there. So you might wonder how these talisins, why don't they attack their own related cells that are around them? So every time we've looked, in no case have we seen a talisin being able to target the producing strain of the bacteria. And what we think is happening, and we don't quite have a perfect explanation for this yet, but every time a talisin shifts in its targeting capability, the LPS also shifts. So the LPS is encoded by a different region of the genome than the talisin, but every time we look in the phylogeny, it's a one-for-one shift. And so we think strains are inherently resistant to the talisins because of the type of LPS they produce. And somehow they're able to change that when they change the targeting of the LPS, targeting of the talisin.
It's so interesting about the differences you see even within a culture. And it shouldn't be a surprise. I mean, any type of organismal biologist or ecologist would say that. But again, it depends on your age group. If you were like raised in the old days, you're very reductionist about it. So it delights me. Let me just put it that way to hear this. And then the final bit that I wanted to bring up with you is the business of using the talisins as protective agents against phytopathogens, which is fascinating.
Yeah, and so we, I didn't really think about this at all until a couple of years ago when we started kind of brainstorming what the potential was. If you think of plants have a very different immune system than animals or a lot of other organisms. And so with our immune systems are responsive, they're adaptive, they can recognize an antigen and then clear out the infection potentially. If you get an infection in a wound or something, our immune system's really good at blocking that infection and clearing it out. There are human immune functions that operate similarly to talisins in terms of how they kill, not necessarily as the same types of structures, but with the same potential to target in a very specific way types of bacteria. I don't know that there's any case of talisins being incorporated into animal genomes or human genomes, but what has happened over time is a lot of other antimicrobial genes have been gathered by organisms like humans, by things like clams, we see them too. Pretty much any animal has types of antimicrobials that have been, where the genes have been transferred from bacteria to the animal genomes, and we use them in things like our tears to actually target and lyse bacteria to prevent infection. So not quite talisins, but other things in a parallel way have happened like that.
Yeah, that's lysozyme, which is absolutely a wonderful thing. Hey, Dave, I had students once who, for an independent project, wanted to test whether or not tears were antimicrobial. And so I said, well, how do you intend to collect tears? And it turned into a whole thing. I was so proud of them.
With plants, they don't have an adaptive immune system in the way that we think of with animals. And so once plants get infected, it's really hard to treat the infection. So we don't, in agriculture, we typically don't treat infections once they happen. We try to prevent them. And so a lot of the agricultural, at least agriculture when I speak of, I'm talking about plants versus the livestock kind of agriculture. But in a lot of plant-based agriculture antibiotics, a lot of it's prophylactic treatments with things like copper or other heavy metals, and sometimes antibiotics, although that's thankfully less used now in terms of just spraying fields with things like streptomycin is frowned upon now versus before. But yeah, so we typically think of preventing infection versus treating it in plants.
I just can't wait until you engineer talocin production on the surface of plant tissues.
Yeah, so the cool thing is, I think we're getting there. There's actually a couple of not for talocins. So talocins are about maybe 10, 12 genes that need to be produced and then exported, and so it gets a little bit hard. There are certain bacteriocins that also interact with the LPS that it's a single gene engineered into plants, and it seems to prevent plant infection in the same way, which is kind of cool.
Oh, I couldn't agree more. Anything we can do to reduce the use of pesticides is just fabulous. No. Well, this has been really interesting, Dave, and I was really glad you took some time. And I haven't even asked about Baltrus farms, as you fully expected I do. And when are you getting emus? That's what everyone wants to know.
I have constantly been not getting emus. So I keep trying.
Well, I think it's wonderful the way that you balance your life together. I like to ask a couple of questions at the end of these, and if you wouldn't mind, I would love to hear you say about the one event that sold you on being a microbiologist.
Yeah, I was trying to think through this. It's a little bit of a longer story than I think a single quick question, but my sister was a microbiologist. She went and got her master's at the Ohio State University. She makes me say it that way. So she got her master's there and then went into industry. And so she makes vaccines. And so I kind of grew up. She was about 12, 13 years. She is 12, 13 years older than me. So I always kind of looked up to her as a role model in terms of what to do. When I was thinking about careers and what I wanted to do, it almost worked in another way, though, because I didn't want to follow her path. I wanted to kind of go on my own path. And so when I went to graduate school, and when I applied to go to graduate school at University of Oregon, I wanted to study ecology in large charismatic megafauna. So I had dreams of like roaming the African savanna and taking notes and chasing down antelope and whatnot. I realized quickly that was not my calling. I am not built for that. But luckily, when I went to start my PhD in grad school, we had rotations. And so I was able to rotate through 3 separate labs. First lab, I did zebrafish genetics in. Second lab, I did Arabidopsis genetics in. And then the third lab, I ended up staying with for my PhD, working on Helicobacter pylori. And so it was a common human gastric pathogen that's well known for kind of being a carcinogen, one of the bacterial carcinogens that's been identified. I was lucky to have 2 great mentors. So I was co-advised by Karen Gilliman and Patrick Phillips, who are spectacular individuals and awesome mentors. They really, they've imparted a lot. Everything I try to do, I kind of learn from them in terms of how to treat people and how to try to meet people where they are in terms of what they want to do research-wise and otherwise. And so they gave me the chance to just develop my own research. I had this crazy idea of, hey, I want to work with Helicobacter, but I want to do experimental evolution. I want to do laboratory passage of Helicobacter and figure out, can we see evolution in action? And really that was triggered. And looking back, like that was the moment that started me down the microbiology path, because I just started a rotation. I was working with bacteria. And I read Rich Lenski's paper, so the 2000 generation lab adaptation paper. And I realized that you could actually do evolutionary experiments in the lab under controlled conditions and measure how things change over time, and measure really easily, like these bacterial cells are going to mutate. We're going to be able to select for it. Things are going to happen in interesting ways. And if you set up the experiments right, you can test interesting hypotheses about evolution in that context. So that's really what got me down the microbiology path, is coming into the right set of labs where I was able to have co-advisors that let me do. Looking back, like, yeah, they definitely guided me towards things that worked. But I came up knowing that I had freedom to kind of ask questions and explore, and they gave me that freedom. And then just building in the lab adaptation work kind of really sparked my interest in microbiology going forward. It's nice to be able to do experiments in the lab that you're measuring evolutionary dynamics, and it takes a couple days sometimes to see that versus many decades for certain organisms. So.
Well, no one can ever say that you're not enthusiastic, because you are. No one can ever say that you don't have a proven track record in mentoring people, because you do. And you have been a fantastic resource to many people, including me. So I want to thank you once again for your willingness to join us on this podcast. And I wish you and your family, and I will also say your colleagues, the very, very best.
Will do. Thank you very much. And then again, best of luck going forward with this podcast. I love being able to talk science, and I love being able to listen to science from a variety of other people talking it too. So this is great.
Well, it's highly therapeutic for me. So thank you very much, Dave. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as usual, wonderful links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Dave Baltrus can be found in the School of Plant Sciences at the University of Arizona. Many, many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next week on Matters Microbial.